This protocol allows researchers to culture stem cell colonies on hydrogels with complete control over both the stiffness of the hydrogel and the geometry of the colonies. The main advantage of this technique is that it allows researchers to culture stem cell colonies in conditions that better mimic the physiological environment than tissue culture plastic. When performing this protocol for the first time, be sure to generate a few extra of every material and use a fluorescent ligand so that you can troubleshoot and optimize at each step. This technique involves multiple steps that involve the precise positioning of various materials. It's much easier to understand the written instructions for this protocol once you've seen it performed.
To begin this procedure, mix PDMS base with PDMS curing agent at a 10:1 ratio by weight and mix thoroughly. Degas this mixture in a desiccator for 30 to 60 minutes or until all of the air bubbles are removed. Slowly and evenly pour the PDMS mixture over the surface of the prepared wafer. Tap the dish on the work surface to release any air bubbles from the surface of the wafer. Bake the PDMS at 70 degrees Celsius for 2 hours and allow it to cool to room temperature. Then cut the PDMS into roughly 10 by 10 mm squares, each containing the features for a single experimental condition. These will be referred to as stamps for the remainder of the protocol. After this, mix PDMS base with PDMS curing agent at a ratio of 8:1 by weight and mix thoroughly. Degas the mixture in a desiccator for 30 to 60 minutes or until all of the air bubbles are removed. Next, slowly